So right now I'm going to explain the TRP operon, which is used in E. coli. So the TRP operon, its natural state, is turned on, which means that the RNA polymerase can bind to the operator and transcribe all of the 5 genes that are responsible for making tryptophan, which is the product of the TRP operon. The 5 genes are translated to mRNA, which are translated to polypeptides, which form tryptophan. This mechanism is also a negative feedback loop mechanism, though, because in the situation of excess tryptophan, what happens is that the regulatory gene that's before that is before the operator, it produces an inactive repressor usually. What happens when tryptophan is in high levels, though, this repressor becomes an active repressor as tryptophan acts as a co-repressor. So it changes shape and it binds to the operator. So when RNA polymerase tries to bind to the operator, this time it just gets stuck and it can't produce any more tryptophan. That's how this negative feedback loop mechanism works. The TRP operon is a repressible operon. Now I'll be presenting the LAC operon. A LAC operon is an inducible operon. So in its normal state, the regulatory gene LAC1 will produce an active repressor which binds to the operator and doesn't allow the RNA polymerase to continue transcribing all of the genes. This means that beta-galactosidase, permease, and transacetylase are not produced. So what happens, though, is that beta-galactosidase is necessary for the decomposition of lactose. So when a lactose becomes present in the system, a protein called allolactase is now present. So what happens is that allolactase binds the active repressor and makes it inactive so that it can no longer bind to the operator. So this means that the RNA polymerase now goes through and it goes to the mRNA and it turns into beta-galactosidase, which then in turn breaks down the lactose. This only happens because allolactase is only in the actual system when lactose is present. This is how beta-galactosidase increases 1,000-fold in just 15 minutes to degrade lactose. So now that I've explained 2 types of negative gene regulation mechanisms, I'm going to explain a positive gene regulation mechanism. This one specifically uses CAP, or catabolite activator protein, which acts as an activator in this situation. So this is useful because when E. coli is in the presence of both glucose and lactose, it likes to use glucose, but it will switch to lactose as a source of energy when glucose is in short supply, and this is the reason how it's able to do that. When glucose is highly present, then cyclic AMP is in short supply. When glucose is scarce and when lactose needs to be broken down and catalyzed, then cyclic AMP is very abundant, and it binds to CAP, and this binds to the CRP binding site. So this protein makes it much easier for the RNA polymerase to actually bind to the operator and transcribe the genes. Without CAP, it's still possible to bind, but it's much less likely for the polymerase to bind to the actual sequence. So this is positive gene regulation because this happens because it only transcribes when CAMP is in high concentrations. So now I'm going to present RNA splicing. So in a normal genetic sequence, there's several non-coding and coding regions of the sequence. The non-coding regions are called introns and the coding regions are called exons. And the introns are not used in the actual expression of proteins, so they have to be removed. And to remove these, that's what spliceosomes and RNA splicing is all for. So, so what happens first is a 5' enzymes add a 5' cap and a poly A tail. A 5' cap to the 5' side of the sequence and a poly A tail to the 3' side. So the 5' cap serves two purposes. First of all, it protects the sequence from degradation, and second, it allows a spot for the ribosomes to actually bind to and transcribe the sequence. The poly A tail also prevents degradation of the sequence, and it also, as well as it allows the sequence to get past the nuclear pores. So what happens now is after the caps have been added, other proteins and SNRMRPs, which are small nuclear ribonucleic acid proteins, are all these three are all added together to form the spliceosome complex. So within the spliceosome complex, the introns are excised out, and the exons are spliced together. And then what comes out is the final actual mRNA that will be used to transfer the proteins. What comes out is the introns, which eventually degrade in the cytoplasm and other spliceosome components. This is how RNA splicing is done.